Cardiovascular physiology, where we're going to look at heart functions, the regulation of these functions, and these controls of the functions over the next two slideshows. So the first thing we have to remember is that there are two distinct types of tissue within the heart that are going to relate to its overall function. One is the myocardium. The other one is what's referred to as the conductive pathway. The conductive pathway are either specialized myocardial cells or specialized connective tissue cells that behave like neurons, meaning they conduct a signal for a contraction to occur, or they conduct an action potential from the SA node through the remainder of the heart, so that each section of the heart will contract in unison. These conductive pathways establish the autoregulatory and autorhythmicity of the heart itself. They do not require nervous system intervention in order to function. However, there is some modulation by the autonomic nervous system, either through epi-norepi or acetylcholine activation. This is primarily done by changing what is usually referred to as vagal tone or autonomic tone, which really is talking about how much acetylcholine happens to be being expressed and exposed to the SA node more than actual change in epi and norepi. However, during a sympathetic nervous system stimulus, epi and norepi will both be released into circulation. The epi and the norepi ends up allowing for an easier rate at which threshold can be reached, both within the pacemaker cells as well as in the conductive pathway cells, whereas acetylcholine tends to make it a little bit harder to do that based off of ease opening and closing ion channels, particularly ligand-gated ion channels. What this does is this allows for a greater with epi and norepi, or a lesser with acetylcholine, force of contraction, not just because of an ease of reaching threshold, but the amount of tissue that will be contracting and the rate at which that contraction occurs. Now, what we have to remember is that the heart itself within the myocardium does not contract to tetany, nor does it contract based off of a Trepp effect, meaning it has to go through a full refractory period before it will contract again. So let's take a look at this pathway of conduction through the heart. The pathway of conduction through the heart will start in the sinoatrial node of the SA node, and then will be conducted through the atria via what's usually referred to as the atrial pathway and the intranodal pathway. The intranodal pathway is the connection between the SA node and a second nodal branch of pacemaker cells known as the AV node, the atrioventricular node. This AV node allows for a pause within the pathway before we get continuation of the conduction through what's usually referred to as the bundle of His, through the bundle branches, into the Purkinje fibers of the ventricles. So let's take a look at how this conduction occurs. So what we end up having leaking of leaking of potassium ions from the SA node, which causes movement of sodium into it, which initiates a wave of electrical activity through the atrium into the AV node, which will then collect the electrical signal and pass along the bundle of His, indicated here as the AV bundle, which heads through the ventricular septum. At this point in time, the atriums have contracted. We then get passing of the signal through the bundle of His to the Purkinje fibers, which wrap from the apex up to the lateral margin of the heart. This causes a wave of depolarization followed by a wave of contraction that mirrors how depolarization will pass. What this ultimately does is determine the intrinsic rate of contraction. That is, what is the physiological basis for contraction based solely on depolarization. This will be affected by stress hormones as well as autonomic tone or vagal nervous input onto the SA node. The pathway of conduction will determine the path of contraction through the heart based off of the spread of depolarization along the conductive pathway. The rate of depolarization and the rate of passage through the pathway will determine the rate of contractions and the overall heart rate of the individual. So let's talk about ways to determine this pathway of contraction. The first way of determining this is with what's referred to as the electrocardiogram. The electrocardiogram is the ability to see the electrical release of energy from the heart through what is usually referred to as the PQRST wave. This electrocardiogram shows the pattern of contraction based on the electrical activity. So if we're to look at it, we have the electrical activity occurring. Note the very short periods of time here, the P, the Q, RS, and then finally the T. And note what's occurring within the heart, where the P is going to be the indication of the atrial depolarization, whereas the QRS is going to be the ventricular depolarization, and the T is the ventricular repolarization. This electrical signal allows us to record the electrical output within those three distinct phases, the P wave, the QRS wave, and the T wave, as well as two distinct intervals, the PR interval and the QT interval. Each one of the phases and each one of the intervals will give us a distinct phenomenon occurring either electrically or kinematically within the heart. So if we look at what's going on within the PQRST wave, we have atrial depolarization occurring at the P wave and atrial contraction occurring within the PR interval. We have ventricular depolarization occurring at the QRS with atrial repolarization occurring. We cannot electrically see the atrial repolarization because it's hidden by the QRS formation of the ventricular depolarization. We have ventricular contraction occurring within the QT interval, followed by ventricular repolarization occurring at the T wave. We then have what's usually referred to as sequestration, which is then followed by another P wave. This electrical output and the full refractory is the reason why we cannot have a TREP and we cannot have tetany of the heart. We can diminish the rate that this takes place, meaning that it can occur faster or it can occur slower, and we can diminish the time between the T and the P. However, we have to go through all phases within the heart rates, within the cardiac cycle, within the heart rate. So let's take a look at this in terms of the schematics. We have our atrial depolarization, our ventricular depolarization, and our ventricular repolarization. Here's the heart. What we're looking at here is we're looking at the propagation and the continuation of electrical signal and kinematic function. The way in which we measure this is with external electrodes. We place external electrodes in such a way as to form an electrical triangle around the heart so that we can see the P, Q, R, S, and T waves of the ECG. Note the various complexes and note the various intervals. Note how each overlays a different function and a different pattern of electrical activity within the heart. Here's the P wave. Note how we have depolarization occurring at the SA node prior to the initial P segment of the wave. We then have the P wave, which is met by the spreading of the depolarization wave through the atria, leading to the atrial systole, the contraction of the atrium. Within this PR segment, we're going to have the small pause at the AV node. This small pause allows for full atrial systole before the initial contraction will occur in the ventricles. It also allows for the depolarization wave to spread through the bundle His into the Purkinje fibers so that contraction will initiate at the apex of the ventricles or the apex of the heart and work towards the superior medial margins during the systole of ventricular contraction. This is then followed by a relaxation and repolarization of the tissue within the T wave. Here is the relaxation, the diastole, followed by the repolarization in the T wave. Once again, we're not going to have a large change in timing within the cycle. We may have some timing reducing the TP intervals, and we may have some differences in timing between P and P, as in the next contraction cycle. So within this contraction cycle and depolarization rate, what's the timing issues? As in, how long does it take? From SA node firing, meaning depolarization, to full heart contraction, we're looking at about 300 milliseconds, or about 0.3 of a second. The intrinsic firing rate, the rate based strictly off of depolarization, gives us a contraction cycle every 900 milliseconds to 1.2 seconds, which gives us that number of norm heart rate being about 60 beats or 60 contractions per minute. The other way that we can determine this pathway of conduction is based off of listening to the fluid movements through the valves of the heart. When we're listening to these sounds, what we're listening to is turbulence around the valves. The fluid as it moves is moving based on what's known as laminar flow, meaning it's flowing very smoothly. Every time we have a contraction, we get a turbulence within that laminar flow. That turbulence causes sound waves to occur, and what we actually hear are those turbulent sound waves. If you follow the link on the bottom of this page, it will take you to a website where you can actually listen to various types of heart sounds, which we'll also listen to in lab. The principal heart sounds are usually referred to as S1, S2. These are the lubs and the dubs. The lubs is the sound that is associated with ventricular contraction or the sound of flow around the AV valve closure and the semilunar valve opening. It's associated with the expulsion of blood and systole of the ventricles. The dub sound is the sound of ventricular diastole. It's usually associated with sounds of the fluid around the semilunar valves being closed and the AV valves being open. Along with that, there are two other types of heart sounds. There's one sound referred to as a murmur, which is usually an S3 sound, and a gallop, which is usually an S4 sound. Murmurs may or may not actually be pathophysiologic, which means they may or may not actually be problematic. In fact, a vast majority of people walk around with some degree of murmuring occurring. A murmur is simply a secondary sound due to an incomplete closure of the valves of the heart. What this does is this increases the turbulence, and by increasing the turbulence, it increases the sound wave. A gallop, on the other hand, is an irregular sound indicating a secondary contraction. The secondary contraction is due to a backflow issue during ventricular filling. Gallops are always pathological in nature and are an indication of ventricular issues, most notably ventricular hypertrophication, meaning that we are not ejecting enough blood to overcome the peripheral resistance to flow, causing a secondary contraction to occur in between ventricular systole and ventricular diastole. The next segment of stuff we will look at the issue of the rhythmicity of the heart rate, as well as heart rate norms for the population.